Okay, so on Monday I send you the microbe of the week question, which is can bacterial endospores survive in outer space? And the answer is yes, they can, because they can survive cold temperature and they can survive being in a vacuum. And we know this because endospores have been left outside in space on the International Space Station for different times. I think a few people sent in reports of that experiment. It lasted 1.5 years, and it was possible to activate and obtain live bacteria after 1.5 years outside the spaceship. Other people that gave interesting answers were things like there was an Apollo mission picked up some kind of something that was left on the moon from a previous mission, and when they got back, they found that endospores had survived being on the moon. So that was quite an interesting thing to find out. Now, the most difficult thing for bacteria to survive, for endospores to survive in space, is UV radiation. That's the most harmful of deep space conditions. So if they're shielded from direct light, I guess, then they can survive for a very long time. And that means that probably if they are in a rock or something like that that's floating in space, then they may be able to survive for at least several years. Who knows, maybe even more. So the second part of the question was, could they survive an interplanetary journey? Most people thought yes. I think it's quite reasonable. Interstellar, I mean, there's a couple of people who sent me the calculations of how far that actually is and how long the journey time would be. And I don't remember. So what is it, in tens of thousands of years or something, or is it more? Somebody who sent a nice calculation. No? Don't want to tell us? No? Well, anyway, it's going to be so long. It's going to take so long. People thought it was going to be unlikely. Okay? And another thing that was pointed out in one of the answers was that, yeah, okay, you might make an interstellar journey, but of course, once the endospores arrive on another planet, they have to find good conditions that will activate them and germinate them. I thought that was a good point as well, because most spore-forming bacteria are heterotrophs. They need organic carbon, and this is what's going to induce germination. So if they land on a planet with no life, that's going to be difficult for them. But there are some endospore-forming bacteria which are autotrophs. They can make their own organic carbon. So maybe some bacteria could actually colonize and start life on other planets. So that was kind of the point of that question. So thanks everyone who sent answers, and I enjoyed reading them. Now today we're going to look at identification and phylogeny of bacteria. Perhaps it's going to be a little bit shorter than the other lectures, so I might finish after about an hour. I probably won't take the full time slot today. Okay, so how do we identify different species of bacteria? Because you look at them in a microscope and they look pretty similar. And the question is really, are they all identical or are they different? Do they have different characteristics? Are they different species? And there are two approaches to bacterial classification and identification which coexist. So before molecular biology had been invented, microbiologists used phenotypic classification to identify different species of bacteria and to construct phylogenies. Okay, grouping similar species together in a genus, similar genera in a family, similar families in an order, and so on, in order to kind of fit bacterial phylogeny into this very kind of Linnaean scheme where you have, kingdoms, phyla, class, order, family, genus, species. And the person really who did the most work about this was a microbiologist called Berge, who published his Manual of Systematic Bacteriology. I think the first edition was before the Second World War, and now it's in, I don't know, 6th or 7th edition, something like that. So this is the Bible for bacterial classification. So this work's been going on for a long time. Bacterial, orders, families were defined on the basis of some kind of phenotypic criteria that the different bacteria share within that group. And since then, we've got molecular phylogeny. So this is based on sequence information of different bacterial genes. So this is a different way a different approach to try and classify bacteria. And it kind of has been grafted on top of the existing phonetic classification. Most of the time it works, but as we'll see, sometimes it causes problems, especially in the higher levels of this classification. And when we get to the end of the lecture today, I think we're going to end up with a discussion about what does that really mean, and can we actually apply a kind of standard phylogenetic model to bacteria or not? And that's the kind of question that has become necessary because of the inconsistencies in molecular phylogeny. So the first part of the lecture is going to be about this: identification and classification with standard microbiological techniques. This is what people use most of the time in the lab. And then the second part of the lecture will be concerning molecular approaches. Okay, so the phonetic approach is you're trying to describe the phenotype of your organism and define phenotypic characteristics that can differentiate one bacterium from another. So it starts out with morphology, visible characteristics. So you look in the microscope, the very first thing you're going to look at is gram-positive, gram-negative, bacilli, cocci, vibrio shape, diplococcus, streptococcus, this kind of thing. Presence or absence of flagella. Colony morphology, what does the bacterium look like when it's in the Petri dish? That can be an important element of identification, especially for something like Bacillus. Okay, Bacillus subtilis has got kind of very characteristic flat colonies. If you see gram-positive rods, they look very long and rectangular, and look at the colony, that's going to be enough to give you a pretty good guess at the identification. Don't need to do anything else. Then, of course, endospores, whether they're present or not, and where they're located in the cell. So to distinguish Bacillus from Clostridium, you look at the type of endospores. Okay, so that's the first thing you can do, but it's relatively limited. It's very, very useful because it's fast and can give you a general idea of what kind of bacterium you have. But you don't have very many characteristics here. Okay, so you've got, this is binary, you have two categories. This cell shape, I know you might be getting onto 4 or 5 really different types of cell shape, flagella or not, the colony. So if you divide all this up, you're talking about, I don't know, 20 or 30 different groups you can define here. But that's going to be quite low resolution, right? So that's not enough to identify bacteria. What you need to do is look for different types of characteristics. And these aren't going to be morphological. That's all you've got here. They're going to be based pretty much on biochemistry. So that is, what is the general nutritional type of these bacteria? Are they photosynthetic? Are they chemotrophs, autotrophs, this kind of thing? What can they use as a source of carbon and nitrogen? Do they ferment glucose? Can they ferment maltose, rhabdose, other sugars? Can they use inorganic nitrogen? Do they need an organic nitrogen source? Similarly, oxygen relationships, that means are your bacteria aerobic, anaerobic, or facultatively anaerobic? Okay, facultative anaerobe can use oxygen, but it can also ferment. Mechanisms of energy conversion, well, that's a similar kind of thing to the general nutritional type. That is, are they photosynthetic? Where do they get their energy from? Do they get their energy from an organic source or an inorganic source? pH, temperature range. Okay, so you're going to talk about whether they're acidophiles, alkalophilic, temperature range. Okay, so if they grow at room temperature to about 40 degrees, it's mesophils. Above 40 degrees, you're talking about thermophilic bacteria. Things that can grow below 10 degrees are psychrophilic. So that's going to give you a whole bunch of information about the growth characteristics and the nutritional characteristics of the bacterium. That's useful information. And then finally, if you're talking about pathogens or symbiotes, then you'll have the interactions with other organisms that are important. So host range. Mycobacterium bovis infects cows. Mycobacterium tuberculosis infects human beings. This is a way to distinguish these two species. So you've got a range of phenotypic characteristics that are going to be present or absent, positive or minus for each sugar or each biochemical test. And what you want to do is try and combine them in a way that will help you get some idea about the identification of the bacteria. So this is what you call an identification key for non-fastidious aerobes. Non-fastidious, that means that they will grow on a rich culture medium, but it does not really have very, very specific supplements. So if you start off, this is what you'll be doing in the practical labs. You start off with a suspension that's got bacteria in it. You take some out, put it on a Petri dish with a rich medium, LB agar, so it's got yeast extract, peptones, and that's about it. So you've got carbon, organic nitrogen, and a whole bunch of vitamins. Then you start off with colonies, incubate them at 37 degrees in air. So already you're selecting for bacteria that are mesophilic and aerobes. So that's your first level of selection. You're not looking for all bacteria here, okay? Only the ones that can grow under these conditions. Then you pick some colonies and start looking at them. So the first thing you do is a gram stain, plus or minus bacilli or cocci, and then you perform some simple biochemical tests that can be done directly on the colonies. So for cocci, what is useful is to perform a catalase test, just to add some H2O2 onto the colony. If it forms bubbles, then they are catalase positive. So that will help you distinguish Staphylococcus from Streptococcus, Enterococcus, or Micrococcus. Now, Staphylococcus, Micrococcus will be here as well. That gives you some interesting information. Same thing for gram-negatives. Gram-negative cocci are very rare, so basically you'll be looking at Neisseria, probably nothing else. Gram-negative bacilli, there are a lot of them. So you perform another simple test on the colony, an oxidase test, and this will help you distinguish generally what are enterobacteria. Could be several different species too. Oxidase positive gram-negatives. Could be Pseudomonas, Vibrio, Pastorella, this kind of stuff. Now, just with morphology and a couple of simple biochemical tests, you can get information down to a family or a genus level. If you want to identify these bacteria to a species level, then you have to perform a whole battery of biochemical tests. And this is also what you will do in the practical lab. So once you have an indication whether you have Streptococcus, Staphylococcus, Enterobacteria, or non-enterobacteria, gram-negative enterobacteria. No, gram-negative non-enterobacteria. Then you can inoculate a small amount of the culture into these small chambers that have a particular medium. So each one of these little chambers here is going to have a particular, either here it's a particular sugar, and the dye will show you whether the bacterium can ferment this sugar or not. So yellow is a positive result, blue is negative. And up here, this is used for use of different amino acids, this kind of stuff. So each one of these chambers here is one biochemical test. So for each one, you get information, positive or negative, for a particular biochemical characteristic. And then you look them up, and you'll find out whether the biochemical characteristics are specific for Salmonella, E. coli, or a whole bunch of other gram-negative enterobacteria. And that's how you get identification down to the species level. Now, below the species level, you might want to you might want to distinguish between different strains of the same species. So, Escherichia coli, for example, it's a gut bacterium, very common. The vast majority of strains of E. coli are harmless. They are symbiotic, or at least commensal bacteria. However, there are some strains which are pathogenic. And if you are isolating these bacteria, you might want to know whether they're pathogenic or harmless. So you need to be able to identify strains below the species level, so different variants or subspecies. Now, sometimes biochemical markers can still be useful. For ability to ferment a particular sugar or resist some kind of toxic material. That can be useful. Okay, the example for this that you do in the TD classes is something to do with resistance of telluric acid. So some E. coli strains have the ability to resist this toxic metal, and that's useful for a screening test. Now, most of the time, what is going to be useful for identifying strains is serology. So you want to use antibodies that are specific for different surface antigens to distinguish between different strains of coli or different strains of Salmonella or different strains of Streptococcus. And as we saw before, the antigens that are going to be recognized by the antibodies are either capsule antigen, flagellar antigens, or LPS, the O repeats. And really the most widespread kind of test or technique for doing this is by performing an agglutination test. Okay, so how does that work? Okay. let's just imagine we got two different strains of bacterium here. And let's imagine we have two different types of O antigen. This one here has got just, I don't know, let's say 1 sugar, 4 copies of it, repeated lots of times. So let's make it like this. Then it's going to be repeated. And this one has a different O antigen repeat. So you get a suspension of these bacteria and you mix them up with latex beads. That are coated with antibodies. Now these antibodies, let's imagine they're specific for this O antigen repeat. When you mix these latex beads up with a suspension of these bacteria, then what is going to happen is that the antibodies will bind to these bacterial cells and it's all going to kind of agglutinate, form a mass. So when you look at this suspension on a glass slide, you put a drop of the latex beads and a drop of the bacterial suspension together. You leave it 15-20 minutes at room temperature. When you look at it, you'll see that you have lots of big, okay. Maybe I should try and make an effort to draw this a little bit better. Okay, here's your slide. You've got a kind of drop of the latex beads. This is this one. This is this one. Here, this O antigen is not recognized by the antibody. So all of the beads and all of the bacteria will just stay in suspension. Whereas here, these antibodies recognize this O antigen and it will form a kind of big agglutinated mass. So you'll see something kind of like this. This is visible. Or you can just see it on a kind of very low resolution microscope, like a 4x binocular loop. So you get a visible result after a short time, and it tells you that this bacterium is a, okay, whatever the serotype it is, could be A, B, C, D, or whatever, depending on how the classification works. So this is the, like we call this the A serotype, and this is going to be the B serotype. So if you have a suspension of bacteria, then you perform this test with a whole bunch of different latex beads conjugated to different antibodies. So this is the antibody that recognizes A. So you would have beads with anti-A, anti-B, anti-C, anti-D, and you would incubate your bacteria with this whole panel, and then you get one of them will be positive. And they say, okay, that's it, that's my serotype. My serotype for this one is E, or whatever it is. So you do this test with antibodies specific for different capsule antigens, different flagellar antigens, and different LPS antigens. And then you'll have a serotype for the capsule, the flagellum, and for the LPS. Actually, these aren't really used as letters. They're used as numbers, to be honest. So for E. coli, you might have H70157. Something like that. Okay, so that's serological typing. Now, molecular genetics. This is really the first case where we're going to use it, is to identify subspecies and individual strains. All right, the first thing perhaps I'll talk about is genetic fingerprinting. So many bacteria have got repetitive sequences scattered throughout their genome. So that's what these are, okay? Rep, Box, and Eric. They're particular sequences that are somewhere in the genome of bacteria. So Rep, I have to have this one written down because I don't know. It's called repetitive extragenic palindromes. And Eric is enterobacterial repetitive intergenic consensus. So if you kind of imagine the bacterial chromosomes like this, okay? And you have these repetitive sequences scattered around. However, from one bacterium to another, the distribution is not going to be the same. These are mobile elements of DNA. They can easily change their location in the genome. So what you do is you take DNA primers that are specific for these repeated sequences and use them for PCR. So here you'll get PCR products that go like this. And you migrate them on a gel. So you'll have, I don't know, one really short one here, one slightly bigger one bigger one, and one really, really bigger one. Whereas here, you'll have PCR products that are like this, okay? Imagine this is an agarose gel. The further you migrate, the smaller the product is, okay? So when you look at this is going to give you different kind of a different DNA fingerprint for the two different types of bacteria. And that's how you can distinguish them. So this will be different between different strains of the same species. Mostly, this is useful for gram negatives, okay? But some gram positives have these as well, all right? So Streptococcus has this one. They're very frequent in gram negatives. That's how genetic fingerprinting works. MLST is for multiple locus sequence typing. That works in kind of a different way. Here we're not talking about repetitive elements of DNA, we're talking about different genes that are present in both the bacterial species you want to compare. So what you'll do is sequence the DNA for genes A, B, and C for these two bacteria, and then align the sequences. So you'll have a whole bunch of DNA sequence, and then from time to time, you might have some differences. And then you can use these differences in DNA sequence to construct a phylogenetic tree. And this will show you whether these two species are the same, or two subtypes are the same strain, two isolates are the same strain, or if they're different, they can tell you how different they are, where they go in terms of phylogenetics. Now, of course, what level of phylogenetic information you get depends on what gene you're studying. If you want to look at different strains of the same species, you want to use a gene that is quite variable, not highly conserved. if you want to look at a kind of higher level phylogenetics, you need to look at very, very conserved genes. Okay, so MLST, you want to kind of choose what level of the choice of genes you look at depends on what kind of conclusions you can draw. Actually, that's the other way around. The kind of conclusions you can draw depends on the genes that you choose. Okay. So that was a kind of slight intrusion of molecular techniques for identification. Now, just forget that for a minute. Imagine we don't have any sequence data or genetic fingerprinting or anything like that. Just think back to this phonetic tests for biochemistry, serotype, this kind of thing. So each of these biochemical tests is a characteristic that's present or absent in the bacteria that you want to identify. So we started off using these characteristics as an identification key. This is just practical use when you have bacteria, you want to identify them. First you do gram stain, then you do some simple tests, then you do some more complicated tests, you get the identification. Now the next thing to do is to try and use this information to construct phylogenies. And that will tell you which groups of bacteria are related in evolutionary terms. So what's been done in the past, really, perhaps not so much anymore, is to try and combine all of this phenotypic information into a kind of simple matching coefficients and perform what's called numerical taxonomy. So for each character that you have, okay, ferments glucose, ferments rhabdose, ferments maltose, this kind of thing, can use sulfate as an electron donor. It's going to be, your bacterium can do this or it can't do it. Okay, so if this character is present, you've got 1. If it's absent, it's got 0. And then for each pair of bacteria, you'll try and find out how much they match. Okay, so for each character, do they share it? They both have it. Okay, you might say, okay, let's say we're looking at 20 characters here. We use 20 characteristics for our identification. So for our 2 bacteria, you might say, okay, in 10 cases, the character is present in both of them. In 4 cases, it's present in bacterium X, it's absent in bacterium Y. In 2 cases, it's present in Y, it's absent in X, and in 4 cases, it's absent in both of them. So we use these numbers and then calculate some kind of matching coefficient, either the simple matching coefficient, so that's the number they have in common divided by the total. So in here, we've got like 14 points that are in common out of a total of 20. So in our example, this would be 0.7. Sometimes that can be a problem because here you're defining similarity by characteristics that these bacteria both lack. This can be very risky because 2 bacteria that are very, very different, they might just both lack the same thing because it's only present in a whole bunch of other specific families of bacteria. So in order to avoid falling into this kind of, and that can lead to artifacts in your phylogenetic tree. So in order to try and avoid this, there's another kind of coefficient you can use, the Jacquard coefficient, which kind of just excludes this data here. So we're only going to consider characteristics that are present in at least one of the 2 strains that we are comparing. So here we would have like 10 over 16. Which is 5 over 8, 0.625, right? Okay, so for each pair, you work out these coefficients, the matching coefficients. And then what you do is you construct a kind of similarity matrix. So you've got all the different bacteria that you want to compare. Each square in this matrix is the matching coefficient between these two bacteria. Obviously, 1 compared to itself, 2 compared to itself, 3 compared along the diagonal, they're 100% identical. And of course, the closer they are phylogenetically, the higher this number is going to be. So you've kind of reduced all of this phenotypic information into a degree of similarity between two strains. And then you can use this to construct cladograms, phylogenetic trees, by, what was the stuff we did last year in mechanisms of evolution? I only know that during the second semester. I just don't remember it further. What are the types of methods we're going to use here? Anybody remember? Maximum parsimony, neighbor joining. What's the other? It's got something to do with arithmetic means, right? Oh, yeah. Unweighted paired group method with arithmetic means. Yes. Okay. So you use these kind of distance methods to get the phylogenetic tree. Actually, you won't be using this. You have numerical data, so you use distance methods. Yes. Okay. What else did I need to say about this? Yeah, so it's generally considered that if you have a value, that's if your matching coefficient is greater than 0.8, then you have two strains which have the same are of the same species. So here, 1 and 2, that would be the same species. Afterwards, of course, it's very, very difficult to tell. Are you going to say this is the same genus? This is the same family? Because the number of levels in the cladogram doesn't necessarily correspond to the number of levels in the Linnaean system. Okay, so if your matching coefficient is greater than 0.8, generally speaking, they would be bacteria considered to be the same species. Okay, so that's how phenotypic information is converted into phylogenies for bacteria. Or at least that's the way it worked until maybe up until 20 years ago. And of course, the big problem is the more the species you have to classify, you need to have a whole load of different characteristics. So it becomes very, very labor intensive to get this information. So here's a paper that was published about 20 years ago to try and determine the phylogeny of different species of Neisseria. So here are all the different bacterial species, different strains that they used. And here are all the different phenotypic tests. So they have a battery of 31 tests here. They performed on all of these different strains of bacteria. Then they got, the matching coefficient for each pair, and then they use them to construct this tree. And it kind of works. You've got the gonorrhea type A, gonorrhea type B. They form two clades. Then you've got the meningitidis group here, meningococcus A and meningococcus B. And then other types of Neisseria are further up the phylogenetic tree. So that was it. 20 years ago, they used this technique to give the definitive phylogenetic tree of Neisseria. So it can be done, but the more species you have, the more labor intensive it becomes. So that was up until about 20 years ago. Bacterial phylogeny was done uniquely with this method. Oh, not uniquely, but it was still being done with a kind of phonetic methods. Since then, and even before, in fact, people have started to use molecular methods because it's just so much more convenient and you can do it on anything. One of the big problems with the phonetic methods is first you have to be able to culture the bacteria. Otherwise, you just can't get any information at all. Most bacteria that are present in the world have not been cultivated yet. So extremophiles, this kind of thing, it's going to be difficult to determine the culture conditions that allow these things to grow. Bacteria that live down at the bottom of the ocean, not very easy to culture them. Whereas if you can extract DNA from a sample, even if you can't culture the bacteria, then you can get some kind of information about what they are. And these are different kind of approaches. So the oldest ones, genome hybridization, extract the chromosomal DNA from one strain from another strain, hybridize it together, see how much percentage similarity they have. So that kind of works for genus and species. But of course, you have to have a pure culture of bacteria to be able to do this in the first place. The next one, these are ones we've seen before, okay? MLST, multiple locus sequence typing, and genetic fingerprinting. Here you can distinguish different genera within a family, different species, and different strains within a species. As we'll see, at a higher level, okay, the genetic fingerprinting is not applicable to higher level classification. The repeated sequences that you use for this, like rep, box, this kind of stuff, they're too variable to be useful for this higher level classification. MLST depends on what genes you look at. If you look at very, very highly conserved genes, this could still be okay. And the one that is perhaps the most widely used, at least as a first kind of approach, is to use the sequence of the small RNA subunit of small subunit of ribosomal RNA, which is 16S rRNA in bacteria. And this is for higher level classification. So let's see how they shape up. I might need to zoom this a little bit. So Neisseria, same genus of bacteria as we saw for the phonetic classification. This was published in 2012. Someone who tried to establish the phylogeny of Neisseria, different Neisseria species, looking at the sequences of 53 different ribosomal protein genes. And they also sequenced the whole genome to find out whether you actually get any new information here or whether you can just use the MLST. So let's zoom in a little bit because it would be very difficult to see otherwise. Okay, so on the right, this is the phonetic classification for from all those 31 phenotypic markers. This is the molecular biology classification. And you actually get quite a good correlation between the two. Okay, so this is the gonorrhea group, gonorrhea A, gonorrhea B. This is that clade. Higher up, you have the meningitis group. Okay, this is what they have here. And then you have the further up in the tree, you've got the polysaccharia group. Okay, you have the same thing here. And then you've got lactamica, which here is the first branch that comes out from the root of the tree. In this case, they have looked at more different groups. So there are some inconsistencies because here they've got cinerea in terms of phenotype in biochemical markers. It's in amongst the meningitidis, Neisseria. But in molecular biology terms, it's nowhere near there. Okay, so mostly it's the two trees you get are consistent. And if you have big inconsistencies, okay, like this one, I would generally say most people accept that the tree based on molecular biology is probably a better reflection of the evolutionary relationships than the phenotypic stuff. Okay. So that works within a genus. Multiple locus sequence typing within a genus to classify different species of bacteria. How about going up in the hierarchy? Can you use MLST to classify different genera within a family? So this is an article from 2012. Once again, ribosomal protein sequences were compared to generate this tree. And this is the Bacillales order. Okay. And generally speaking, it works. Okay, so you have all the Streptococcus, they're all together, they form a clade. Staphylococcus, that forms a clade. Lactobacillus, they form a clade. Listeria, or they form a clade. But there are some interesting aspects here. Okay, so Streptococcus and Staphylococcus, in terms of molecular phylogeny, are really very different. Because if you were just looking at them under the microscope, you'd think, oh, they're quite similar things. In fact, in evolutionary terms, no, they're not. They're quite different. And Staphylococcus is going to be closer to Bacillus than it is to Streptococcus. Another thing that you might notice is that there's one big inconsistency here, with some Bacillus species are up here in this clade, and most of them are here. So Bacillus is a genus which is identified on its morphology, gram-positive rods that form spores in a particular place. So perhaps that's a little bit misleading. And we have really two groups that share this kind of morphology. And if you're thinking about cladistic terms, okay, you've got rod-shaped spore forming, and then, it's being lost. They lose the capacity to form spores here for Listeria. Listeria doesn't form spores. So there are some inconsistencies here with what you might think, in terms of classical bacterial phylogeny. But the big difference between Lactobacillus and Bacillus, that's maintained. Because this Lactobacillus group, they're known as non-spore forming. Oh, in terms of, gaining and losing characters, okay? Try and think back to last week. What has happened at these 2 points in the cladogram? They gain or lose something. Yes. They're cocci, so they lose. MBL, the actin homologue that is responsible for the elongated shape. So within this group, the archaic state is bacilli, they had it. And then these two groups probably lost it independently. And that's why they're coxy. They just lost that gene or lost its expression or its function. Okay, so within a family or within an order, MLST seems to be working pretty well. It defines groups that biologically have meaning, okay, and it mostly conforms to what we already knew about bacterial classification. And the inconsistencies are probably quite interesting and telling us something new. So next thing they did was to try and use the same technique to classify every single species of bacterium. So they did this with, I don't know, several hundred species of bacterium and tried to get a universal phylogenetic tree of eubacteria. Okay, not all prokaryotes, not the whole of life, just standard eubacteria. And sometimes it works, for some groups it doesn't work, okay? Several inconsistencies here. So generally speaking, most people would consider the proteobacteria to be a major phylum of the bacterial world. But if you look here, okay, here it works. Beta, gamma, alpha proteobacteria. Ah, no, it doesn't work, look. This is on a major branch. And then you've got the epsilon proteobacteria in the middle here. Most people think that they should be together with the other proteobacteria here. So that doesn't work. And there are other things that are also inconsistent. If you look at Clostridia, now some of them are here on this branch of the phylogenetic tree, and other ones are miles away. And really, this is quite a big difference here. It's not easy to reconcile these inconsistencies with what we know about the biology of these bacterial groups. Okay, some of them work. Okay, the Bacillales, this order here, that works. Okay, and as we saw before, the classification within this order is still okay, but at a higher level, it's not really working very well. So how do we get higher level classification? Well, the thing that people fall back on most of the time is to compare the 16S rRNA sequences. And okay, and it's useful because there are zones of the sequence which are very highly conserved, so you can use the same nucleic acid primers to amplify up the rRNA sequence the middle part, which is less conserved, and then you can use this variable regions of the 16S rRNA to compare bacterial species. But generally speaking, rRNA is very, very highly conserved. All organisms have it, and so if you take a 2-by-2 comparison, if you've got more than 97% identity, you consider this is the same species. Greater than 95%, the two bacteria are in the same genus. And the less identity you have, okay, the further away these bacteria are in phylogenetic terms. Now, the other advantage that the small subunit of rRNA has is that it's also present not only in eubacteria, but also in archaea and eukaryotes. Interestingly, there are regions that seem to be signatures for what are called the 3 domains of life. So in red here, these are parts of the rRNA structure, 16S rRNA, that are typical for eubacteria, archaea, and eukaryotes. So this thing here, this is eukaryote specific. So this idea that there are 3 domains of life was a big surprise for microbiologists, and it was kind of came from studies by Carl Woese. I think at the end of the 1970s he published the first results comparing eubacteria, archaea, and eukaryotes. So in terms of 16S rRNA sequences, you can define 3 groups: bacteria, archaea, and eukarya. This is the 3-domain model of life. Now that's something that caused, well, it was a big surprise, because just in terms of morphology and the way cells live, people thought, bacteria and archaebacteria, they're quite similar. Okay, so these are what we call prokaryotes. Now some microbiologists say that this 3-domain model means that the term prokaryote doesn't have any biological sense, and we should stop using it because it kind of lumps together the eubacteria and archaea in the same category, and we really shouldn't do that because they're too different. I should say that's a minority view. Most microbiologists, including me, think that these types of organisms share a lot in terms of their basic way of life. They're simple unicellular organisms, no nucleus, no complex internal structure, and that has a big impact on the rest of their biology. So the term prokaryote does make sense in terms of telling us something about the way these organisms are structured and what kind of lifestyles they're going to have. So most people think we should keep the term prokaryote because eukaryotes are very, very different. Now, the other thing that is quite surprising about this is really the length of this branch here, of the eukaryotes. Because in molecular phylogeny, there's this idea that the length of the branch is correlated with the amount of time between different events. So if you look at the origin here, this is the origin of the eukaryote lineage. And this branch length is very, very, very long. And that kind of implies that the eukaryote lineage is extremely ancient. And that kind of would be a problem, because generally we think that the eukaryotes were formed by some kind of fusion event between a proteobacterium, which became the mitochondrion, and some other unidentified partner, which was probably an archaea. But if this is really correct, then that means the eukaryote lineage is really, really, really ancient. So that has been a bit of a headache for microbiologists for a long time, and I think this question has really been resolved this year. There's a paper published earlier in 2015, because, okay, some people have said, right, this idea, this 3-domain system is an artifact. It's an artifact because we don't really have a lot of information about the archaea. So we have a small sample of archaea. We have a large sample of eubacteria and a large sample of eukaryotes. So we don't really know what the diversity of the archaea is. And maybe if we sampled some more archaea, we would find that, in fact, they're a lot more similar to eukaryotes. So that this long branch here is just wrong. It's just an artifact of the sampling that we used of the number of the taxons. So choosing your taxons wisely is very, very important, because if you choose them the wrong way, then you pull out a tree that is wrong. So there's been a bit of an effort to try and look more widely at archaeal sequences and find out and try and find more diversity within archaea, so that we could perhaps get a correct idea of this universal phylogenetic tree. So I was reading this paper yesterday, and in fact I mixed up my timing for today, because I thought I was with another group this morning, and I thought I would see you this afternoon. And I would have time to work this into the lecture and prepare slides, but I didn't. So I'm just going to show you a couple of things from the paper. Okay, there it is. Okay, so let's have a look over here. Okay. Oh, I won't be able to zoom if I do that. Okay. Okay, so this is a sample of bacterial DNA that came from a deep ocean vent. This is Norway, okay, this is Greenland. It comes from here, up near Svalbard, underneath the ocean. So he got a bunch of DNA and managed to get a whole load of sequence information from the DNA that's down at this underwater thermal vent. And they were able to identify a new kind of deep sea, what they call DSAG, so which is deep sea archaeogenomes. And they call them Loki archaeota because this kind of thermal vent forms a structure which is called Loki's castle, and they sampled over here. So they named these archaebacteria after Loki. So they were able to get full sequence information from the Loki bacteria, and this is changing what the archaebacterial phylogenetic tree looks like. So these are the 3 Loki archaea. Here are some other types of archaeobacteria, and here's the kind of base of the tree here. If you put eukaryotes in, we've got eukaryotes as they're just a part of this higher amount of archaeobacterial diversity. So in fact, that means that the tree we had before kind of goes like this: eubacteria, archae, and eubact, and eukaryotes. This is wrong. Okay, once you sample more archaebacteria, it looks like this. You've got archaebacteria with a bunch of things, and one of these branches is the eukaryotes. That's this tree here, okay? And the other interesting thing about this paper is that is when they looked at, okay, there are lots of trees. The other interesting thing here is another puzzle about the origin of eukaryotes is that eukaryotic cells have got a lot of genes that seem to be present only in eukaryotes, not present in eubacteria, not present in archaebacteria. So where did they come from? This has always been a bit of a mystery. Okay. What is very interesting here is that, these are kind of like the different eukaryote-specific protein families here, or different types of proteins. So all of these, they're present in eukaryotes. Now, if they look at these Loki archaea, they have a lot of these things, okay? So that's where they came from. They weren't invented. They didn't come from nowhere. These gene families, and they're involved in things like membrane transport. This kind of stuff is very typical for eukaryotic cells. Bacteria don't do a lot of it, forming vesicles, this kind of stuff. So this paper has really resolved, I think anyway, a long-standing question about, what is the origin of eukaryotic cells, and where all these eukaryote-specific genes came from. So the other partner in this fusion between proteobacteria and an archaeal cell that produced the eukaryotes was probably something in this loci archaeota. So this is the other partner that came to form eukaryotes, or maybe, who knows, higher up in the phylogeny. Okay. Right, so just to kind of, so that was a big surprise from last year. So that's great. That's one problem that's been solved. Other problems still persist, okay, in bacterial phylogeny. One of them is that it's difficult to, the species definition in bacteriology is a little bit difficult to clearly define. There's no clear defining line. Okay, so if you look at the phonetic classification for the matching coefficients, kind of say that if the matching coefficient is greater than 0.8, then two bacteria are in the same species. But you're always going to have cases like it's 0.799 or 0.801 or something like that. And you're going to have to make a choice. Genome hybridization greater than 70% is the same species. 16 srRNA greater than 90% identity. But of course, these are all a little bit arbitrary. And it's a little bit disconnected from the rest of biology. So it's not really, the biological species concept doesn't work for bacteria. Maybe we have to use something like the ecological species concept, which might explain, why bacteria, if you isolate Escherichia coli from my gut and Escherichia coli from the gut of somebody living in Mongolia, still going to be the same bacterial species because they occupy the same ecological niche. Yeah, this one is a kind of a biological species concept says that, okay, bacteria that individuals that reproduce to have a common system of reproduction, they're the same species. But for example, Shigella and Escherichia coli, E. coli, they can conjugate, they can exchange genetic information, but they're considered to be two different species. So probably normally we should say that Shigella should be considered, these should all be grouped together, in fact. But for historical reasons, they stay as two separate species. The other big problem is this one, okay? So when we looked at this higher level classification of all the eubacteria by multiple locus sequence typing, there are a lot of problems. Why do we have these problems? Well, some people say the problem isn't with the biology, it's with our concepts. Okay, so we kind of consider that the history of life is kind of like this. It's a tree structure. You've got the origin of life down here, the universal ancestor cell. And then you get a split off to archaea and eukaryotes and then eubacteria. And then you get two big events of gene transfer with the mitochondria and then the chloroplast being transferred into eukaryotic cells. But apart from that, everything is nice and vertical, with genes being transferred from one generation to the next. And that's what gives you this tree structure. But this researcher, William Ford Doolittle, says, no, we're wrong. This is a wrong way to look at things. There's no point in trying to find the correct tree, the real true tree. This is like looking for the Holy Grail or the Philosopher's Stone or something, because in fact the history of life isn't like that. The history of life is kind of like this. There is a lot of gene transfer events going on at different periods of history. And that means that, your phylogeny might be okay up to about this level. But the further down you go, the messier it gets. And so the idea is that the reason why these higher level trees seem to be inconsistent is because, well, there isn't really any tree. Just in our minds, it's not a real reflection of the evolution of life. So that's quite a depressing thought in that it implies that we aren't really going to be able to get any useful information about the origin of bacteria and the origin of life from a phylogenetic approach. However, maybe things are not so hopeless as that. So if we go back to the 16S rRNA tree for eubacteria here and some archaea that are up here. Does this tree represent something useful for biology? Well, one thing is quite comforting about this is that all the proteobacteria go together. We don't have them scattered around everywhere like we saw in the tree before. And you've got a big difference between gram-negatives, they all go together in the same clade, and gram-positives. So that's quite nice. Okay, we know that it's a very, very basic difference in cell wall structure, and it's comforting to see that this difference comes out in the molecular phylogeny as well. Good. One of the problems, though, is that you look at the branch length here. It's very, very short. So it's difficult to know, of course. And of course, okay, you've got gram-positives here, gram-negatives here. What you might want to find out is what was the archaeal state? What was the archaic state? Were the first bacterial cells gram-positive or gram-negative? You can't tell from just looking at these. You've got to look at the outgroup here, okay? And that's where things get a little bit complicated. So, Deinococcus thermus, they have an outer membrane, but it's no LPS. Chloroflexi, they're gram-positives. Okay. So what it looks like is that gram-positive is the archaic state of this character. Original eubacterial cells were gram-positive. Amongst the high GC gram-positives, you have the mycobacteria, okay, as we saw, or the microbe of the week people saw. These guys also have an outer membrane, but once again, it's different, not the same thing as gram-negatives. So it looks like gram-positive is the primitive state, and the presence of an outer membrane is a derived character. And it seems to have occurred 3 times independently, here, here, and here, to produce an outer membrane. So why? What's the selection pressure that is acting to favor bacteria that develop this outer membrane? Probably it's antibiotics. Antibiotics are produced by other microbes. Most of them are produced by other bacteria. So bacteria are trying to poison each other all the time. And if bacteria can develop an outer membrane and prevent these antibiotics from gaining access to the cell, it's a big selective advantage. So far, so good. I think, okay, gram-positives were the original, it's the original state of bacteria. But Aquifex is gram-negative. What does that mean? Does that mean we're wrong? Bacteria already had a membrane, then they lost it, then they got it back again? Unfortunately, once we get to the root of the tree here, we have a lot of problems because it seems like there was a big lateral gene transfer from proteobacteria to Aquifex. That's why they're gram-negative. Similarly, so this is probably right, and the fact that Aquifex is gram-negative is because of a later lateral gene transfer event. Probably, okay? This is the kind of problem we have. We can never be sure about this stuff. It's just our best guess about what happened in the ancient past. And one last thing, and then I'll stop. That is that if you look at these groups that are really near the base of the tree here, this is what? This is an unrooted tree, but here, this is the last universal common ancestor up here. What are these guys? These are both thermophilic bacteria. They live in hot springs. So this has been used as an indication by some people. Say, okay, that's it. The origin of life is in a hot spring. This is the environment that saw the first cells evolve. Can we be sure? No, we can't be sure. Because when you look at the genome of Thermatoga, once again, it's a kind of hybrid. There's been a lot of lateral gene transfer from an archaea, an archaebacterium, and a firmicute, one of these guys. So the position on the tree might be an artifact of gene transfer from other groups. That's why they go close to the root here, because they've got a gene transfer from archaea. It might not be telling us anything at all about the environment of the origin of life. So once we get up to these very high level areas of bacterial classification, there are a lot of problems. Difficult to resolve, but perhaps with more data, then we can sort out some of these questions. Okay, thank you very much. I think that's about it for today. Probably no microbe of the week today. One a week is enough, right?